Today I'll be presenting about big data, big brain data science, and predictive health analytics. So to begin with, I want to show you a brief outline of what I would be covering in the next 40 to 45 minutes today. First of all, I'll start off with discussing some driving biomedical and health challenges that lead to some of the necessary developments in the data science world and the computational statistics world. So then I would focus on identifying some of the common characteristics that oftentimes describe big brain data. I will explicitly define what I mean by data science and predictive analytics. And then we'll go over several case studies, specifically in the domain of neurodegenerative disorders. I'll show you one cool application of data dashboarding using the SOCR dashboard web app. And then finally, I'll present to you the idea behind a really interesting new methodological development, something called Compressive Big Data Analytics, that is intended to provide a canonical way of mathematically representing, statistically analyzing, and computationally processing large, complex, heterogeneous data sets. So that's the outline. So let's start off with some examples of driving biomedical and health challenges. So this is a recent study that we did a year and a half, 2 years ago, looking at Alzheimer's disease specifically as a component of the vast spectrum of neurodegenerative disorders. So in that specific study, we had about 800 Alzheimer's disease subjects coming from the Alzheimer's Disease Neuroimaging Initiative, a large public-private initiative to aggregate very heterogeneous, multi-source, multi-institutional collection of data for patients, Alzheimer's disease patients, as well as asymptomatic controls, and these individuals that are kind of transitioning from being controls into being patients, something called MCIs, or mildly cognitively impaired individuals. So in this specific study, we looked at the 3 different cohorts, and what we did is we extracted about 80 what we call derived neuroimaging biomarkers, and then additional 80 associated SNPs that were highly associated with the diagnosis. In this case, controls, MCIs, and ADs in that kind of progressive spectrum. So the images on the left here kind of illustrate some of the parcellations that we did for the brain and some of the statistics that we extract, and you can find more about that study in that publication. So there is, the recognition that when you analyze biomedical or health data, there is typically 3 facets of variability that one needs to be aware of. The first one is obviously the phenotype, which is the outward manifestation of the form, shape, size, characterization of the specific individual, if you're doing precision medicine on an individual level, or cohort-based science, atlas-based studies and population-based studies, or you can be doing phenotyping based on various comorbid conditions. In addition to the phenotype, we have obviously the nature or the genome that plays role in everything that we look at in biomedical world. And then there is also the nurture and the environment. Now, across these 3 pillars, I guess, of natural studies, chance plays a big role. So there is random effects, there is noise that needs to be dealt with, and one of on the right side here, I try to indicate this notion that most of the studies effectively resolve into trying to tease out effective biological signal relative to the natural presence of biological noise, as well as noise that's technically introduced by the equipment, by humans, by the processing, etc. So most of the studies that people do effectively revolve around trying to maximize the information content or the signal relative to the corresponding noise that is unavoidable to a large extent. So in this next slide, I'm trying to make the case for why should we look at big data and what is big data and what is standard data or classical or small data. So in this left panel here, I have a representation of a natural process here. So a native process could be, you know, something that's inscribing in this specific case, go blue here with different type of characteristics for these individuals, colors, orientations of these subjects, and so forth. And then on the extreme, on the right-hand side, you have typical what a sample data set represents. You know, it's a snapshot that is pretending to be a proxy of the actual population, which oftentimes is not completely observable through a census. So most of the time, we view these limited process snapshots of the observations, and we try to make the inference using data on the bottom right corner to infer the scripture that's on the bottom left corner. Now, big data is this kind of in-between case where you're collecting, obviously not a census of the population, but you're collecting sufficient amount of large-scale information about the process, looking at different directions, different scale, different modalities, etc. And you're trying to, again, do the same thing. You're trying to infer characterization of computable phenotypes on the actual native process, which is oftentimes not observable in its entirety. So obviously there is pros and cons for looking at sample-based inference statistics versus looking at big data inference, where, you know, there's lots of challenges in terms of the harmonization, the aggregation, the processing, etc. So, by the way, my colleague Crystal, she's going to be reviewing. If anybody has any questions, you can post them in the chat, and towards the end, we'll probably have a chance to go over some questions or comments that may arise during the presentation. So in this next slide, I'm showing you what we call the constructive definition of big biomedical data. So big data obviously was first defined by IBM in terms of the 4 V's about a decade ago. They recognized that the volume, the variety, the speed at which data changes, and the veracity are important characteristics, and they call these 4 V's. Now these 4 V's are nowadays extended to 7 V's and 8 V's. There is more V words that describe the complexities that have to do with big data. Now last year what we did is we tried to come up with a more structured, constructive definition that identifies very specifically what are the challenges and what are the needs for tools, software, and services that are very specific to these, what we call big biomedical data, big brain data, etc. So the size is obviously one of these characteristics. It's one of the paramount characteristics, but we don't stop there. There is complexities with the data that requires special type of data handlers, wranglers, that allow us to manipulate heterogeneous data elements. Then you have data incongruency. The data would not be homologous between subjects, between sites that are acquiring the data, and so forth. So sampling rates. So there's a lot of tools that are necessary and techniques to deal with this incongruency. Another very important characteristics of big data is that it oftentimes arrives from multiple sources. So the transfer, the joint modeling and processing of these disparate elements is a huge barrier currently. Then you can be looking at the same process, the same phenomenon, for example, the human brain at multiple scales. Starting on the top at the macroscopic level, you have typically resolution of the brain at about 1 millimeter, and then you can go down to cryotomographic data where you can have microscopic or mesoscale, and then you can go down to the nanoscale or single channel recordings where you're on the micron level. So you're looking at exactly the same substrate, exactly the same process, but you're looking at different resolutions. So the multi-scale aspect of big brain data is very important. And last but certainly not least is the fact that big data is always incomplete. So mechanisms of dealing with that incompleteness, our group and other groups are developing batch mode techniques, because oftentimes if you have, as soon as you cross the 10 to 100,000 cases, you cannot, it's simply computationally impractical with current technology to do holistic, what's called imputation, to obtain complete snapshots of the data that can be processed using more traditional techniques. So let's go ahead and define specifically what we mean by data science and predictive analytics, because these terms are important. So in my world, data science is defined as this emerging, extremely transdisciplinary area which bridges the theoretical, the computational, the experimental, and then the biosocial or the biomedical areas. Data science deals with these characteristics that I defined earlier, these enormous amounts of complex, incongruent, heterogeneous, dynamically changing data that arrives from multiple streams or multiple sources. Data science always develops methods, tests methods, validates methods, shares methods, tools, services, and capabilities to enable ingestion of these complex data sets and applying completely automated or semi-automated decision support methods and systems. Predictive analytics is the process that we use for utilizing advanced mathematical formalizations, powerful statistical algorithms, and efficient software tools, as well as web services to, number one, represent, number two, interrogate, and number three, interpret complex data sets. So predictive analytics focuses on identifying trends, clusters, motifs, prognostication of process behaviors either across time, space, within range, and outside of the range of the originally observed data. And again, I want to remind you of the little cartoon that I had early on about the complete population data of the Go Blue schematic versus the big data set and the small samples or traditional samples, because this notion of observing data outside of range and inside of the range is very important. So, and by the way, these two definitions come up from an, they're directly coming from an upcoming Springer book on the topic of data science and predictive analytics. So this is a diagram that illustrates a typical process when you're analyzing big brain data. The typical process starts with collecting the data that comes as raw observations, bits, bytes, that are typically aggregated. You have data scrubbing, data cleaning, manipulations, and so forth that need to be conducted first before some kind of an information gets extracted out of these data. For instance, summary statistics. In our case, we take the raw imaging, magnetic resonance imaging scans, for example, and then we remove the non-brain tissue. Then we model the cortical surface of the brain, which is the boundary between the gray matter and the cerebrospinal fluid, using mathematical two-dimensional manifolds. So that's in the information content. And then the next step, the next column here is the knowledge generation phase, at which you begin to look at these models and you obtain summary statistics. So for example, you can be computing surface areas, you can be computing shape indices, you can be computing fractal dimension, you can be computing a variety of morphometry measures for each of these areas. And then based on these, you typically develop learning algorithms or classifiers that can lead to some action that obviously in our world pertains to mediating normal development or various heterogeneous brain disorders. So I want to start with some examples now or case studies. So in the first case study, this is a case study of amyotrophic lateral sclerosis, a very debilitating progressive neurodegenerative disorders. So we wanted to identify predictive mechanisms to detect, track, and prognosticate the progression of AOS across time. Now, although AOS is not extremely common, there is about 30 to 50,000 cases in the US. It is very debilitating. It's also called the Lou Gehrig's disease, and Stephen Hawking is one of those prominent, outspoken, obviously, patients with AOS. So in this specific case, we had a very heterogeneous data set that started, again, over 100 different features were recorded for a number of subjects. They included demographic data, clinical data, what are called AOS, FRS, these amyotrophic lateral sclerosis functional rating scale clinical measures. There were, in addition, adverse events included with some of the patients. And we started with about 8,600 cases. In the process of stratifying the data and manipulating the data to the point where it becomes a single computable object that can be jointly modeled and interrogated, the data was reduced to about 2,400 cases, simply to avoid missingness, to avoid incongruencies, and so forth. And then we looked in this specific case, trying to predict the progression of the disease in terms of monitoring these AOS, FRS metrics, very specifically finding the slope. So we are given the information at baseline, 0 month, and then at month 3, one quarter later, we're given this information and we're asked to predict a 12-month follow-up. Where is the AOS, FRS clinical score for these patients going to be? So we developed this fairly elaborate protocol that's shown in the left that kind of illustrates very concretely how we go from the original observations all the way down to the predictions and then the assessment of these predictions in terms of various quantitative evaluation metrics. The panel on the left kind of shows you the variable importance plot that kind of directly identifies features that are important in that prediction of the 12-month follow-up from the first 3 months of observations. And then the table on the bottom right kind of illustrates 3 different types of classifiers, linear model-based techniques, and then 3 model-free machine learning-based techniques using random forest, Bayesian adaptive regression trees, and a superlearner method. And then the rows in this table kind of illustrate 3 very specific metrics that quantify the performance. One is called the R-square metric that identifies the relative model performance, the root mean square error, and then the correlation between the actual observed AOS FRS slope change between the first 3 months and the 12th month, and the actual predicted by the corresponding method slope change. And you can see that we are reaching about 0.5 or so prediction accuracy. The second neurodegenerative study that I want to point out is a Parkinson's disease study. So there is something very similar for Parkinson's as there is for AOS. There is clinical scores called Uniform Parkinson's Disease Rating Scale assessments. And again, we are trying to predict the progression of the clinical diagnosis of the patients. Can we separate, for instance, patients from age-matched controls with and without using the corresponding UPDRS clinical assessments that the physicians base their diagnosis on. So the table on the bottom kind of illustrates again a little bit about the cohort. There is genetics data, there is imaging data, there is 3 different cohorts, asymptomatic controls or their controls, the patients, the PD patients, and then there is these patients that are in between. They're called SWEDs, subjects without evidence of dopaminergic deficit. So if you look at their imaging scans, the radiologist cannot clearly identify a Parkinson's disease deficit in terms of dopamine change. So for that specific study, we again developed an end-to-end protocol that starts with the data aggregation on the very left. It goes through the data harmonization, aggregation of all these different modalities of the data coming from the multiple sources. They're very incongruent. They cannot be represented in the same functional basis. Then we did the cleaning, the imputation, the wrangling of the data. And then because the cohorts in the previous slide, it was shown that the sizes of the 3 different cohorts that we are studying are not very balanced. So for example, the patients outnumbered controls 3 to 1. There's a potential bias that may be introduced by the patient cohort. So we used a synthetic minority oversampling method, which is a statistical technique that allows us to rebalance approximately, not exactly, but approximately the sample sizes of the 3 cohorts so that we avoid potential bias. And then we did both, just like in the AOS world, we did both model-based, for example, linear model and model-free classification of all the patients, and then we did predictions and we validated the results. The workflow diagram that's in the bottom left corner here shows you the end-to-end protocol. The data goes essentially over 80 to 90 different processing steps to go from its raw aggregated state into some of these cortical surface maps and parcellated maps and statistical signature vectors is what we call these numbers that drive the actual classification at the end. And on the right-hand side, you have, again, a variable importance plot that kind of illustrates which feature, and you can see here there is demographic features like age, there's imaging features like the left superior occipital gyrus surface area is one of these features. You have clinical measures. The UPDRS part 1 was highly predictive of the diagnostic outcome of the subjects, etc. And you can see in the bottom right table, again, some stunning statistics, and these are certainly not surprising. You know, adaptive boosting and support vector machine classifiers were able to discriminate between the different diagnostic cohorts incredibly well. Now, this was simply because we used the actual UPDRS, the Uniform Parkinson's Disease Rating Scale measures, which the physicians themselves used in the determination of a diagnosis. When we exclude these clinical UPDRS biomarkers, our classification goes down to about 80%, which is a substantial decrease. However, it is still pretty impactful and reliable indication that machine learning methods can be used to do fairly powerful predictive analytics. So in this specific case study, just to summarize, we have developed an end-to-end big brain data analytical protocol that can jointly model, represent, and interrogate imaging, genetic, clinical, and demographic data to assess the risk for Parkinson's disease. The source code is available on this GitHub site under the Predictive Big Data Analytics SOCR sub-project. Now let me follow up with something very recent. So this is hot off the press, as they say, although it's not yet peer-reviewed. It is a very, very interesting new study looking at Parkinson's disease patient falls. Can you predict physical injury from falling in Parkinson's disease? So in Parkinson's disease, there is two clinical phenotypes. There is tremor dominance with people that have limbs that basically shake. And the second type is called gait instability and posture instability, or posture instability and gait disorder. So these are freezing of gait and various other disorders. So in this specific case, we were interested in doing three very specific challenges. The first one is we had data from two different sources. Here at the University of Michigan, we have a UDAO Center for Excellence in Parkinson's Disease, a U50 grant funded by the National Institutes of Health, where we're collecting a lot of data on Parkinson's patients. And a colleague of ours at Tel Aviv University collected completely independently a very similar data set, again studying the same cohort, not the same individuals, the same cohort of PD subjects. So the first challenge in this study was to try to harmonize and aggregate this multi-source and complex data set. The second challenge was to identify what are the clinically relevant features that are associated with very specific clinical traits, for example, falls. Okay, and the third one is trying to predict. Now, once you have the feature selection, can you predict falls in this Parkinson's disease using, again, model-based or model-free machine learning methods? So the study design is illustrated in this diagram. First of all, we did this independently study the UDAO University of Michigan data set. We did the feature selection, we did the modeling, we did what's called statistical internal cross-validation of the results. And we did this independently for the two data sets, the Tel Aviv University data set and UDAO. Then in the second phase, we actually aggregated the two data set, and then we did the modeling and the cross-validation on the aggregate data set, as well as we did an external out-of-bag training on one of the data sets and testing or assessment on the other. So the results were fairly encouraging. We have, as I mentioned, missing data is one of these key challenges that characterizes big biomedical data and big brain data in specific. So this diagram on the chart on the left corner here shows you the missing pattern of the data. And on the right, on the top right, I kind of show you some of the differences in four clinically relevant variables like tremor score, posture instability and gait disorder score, and various other metrics between the fallers and the non-fallers indicated with purple and yellow colors. And then one of the big challenges that occurred in this case study, which is certainly very common when you do big biomedical data, is the fact that the data will not be congruent or statistically aggregatable. So the diagram that you see, the histogram plot on the bottom left corner, illustrates the very substantial amount of difference between the distributions of various features between the Michigan and the Tel Aviv data. So everything that exceeds that threshold, that red curve, that I hope is visible here, the horizontal red curve, this is inverse log scale of the statistical significance on a Kolmogorov-Smirnov test between different features. And you can see about half the features are not statistically distinct between the two populations, but a huge number of features are very, very distinct between the two university databases. So how can this be explained? How can this be handled to allow us to literally aggregate and learn from a bigger data set? So okay, so we moved forward. We dealt with these challenges, and I just want to show you some of the results. So we did the feature selection by two different very interesting methods. One is called a random forest feature selection. And you can see in the middle column here the feature name, the variable names, and then right next to it the frequency of how frequently and how common was it, how important was it in the prediction of the clinically relevant outcome, in this case falling or not falling. And then knockoff filtering, the second column in this table on the right, illustrates a controlled variable selection where you have a mechanism of controlling the false positive rate of selecting these features. And you can see that 7 of these features are commonly chosen by the 2 completely independent strategies. So that's a very strong indicator that we might have zeroed down on which observations, which data elements would be highly predictive or associated with false in Parkinson's. The bottom right table that's shown here in the corner illustrates a variety of metrics from model-based logistic regression of identifying followers from non-followers to random forest, adaptive boosting, XG boosting, a number of alternative machine learning-based methods. And you can see that, one of the best classifier, in this case random forest, achieves accuracy of about 80% with a fairly high specificity of about 0.87, and sensitivity not as good, but about 0.7. So this is again fairly encouraging. Now, once you have that, you can obviously make recommendations to physicians, families, caregivers, and so forth about what to watch for or what to look at. Now, in this next case study, we looked at a more generic population, almost looking at the census. So this data comes from the United Kingdom. This is the UK Biobank. In this specific case study, we wanted to discriminate between healthy patients and multiple comorbid patients with multiple comorbid conditions. Now, in the UK Biobank data, what is interesting is that they tried to take a snapshot of the entire population in this case, and this kind of shows a little bit about the data. So over 500,000 cases, over 4,000 features. They do include imaging data, not for all the subjects, but for many subjects, about 10,000 subjects. They have structural and functional imaging and diffusion imaging data. They have genetics data and so forth. So it was a very challenging study. And the URL links that are provided here in the bottom right corner might be useful if you want to follow up with this. This is all public data. You can request data. So just to show you some of the complexities here, right? So we looked at the features, these, literally 4,100 features, and we only looked at the ones that are highly observed. So more than 30% of the cases having observed data. And this is just a plot to illustrate the missingness rate, okay? So it turns out that in our situation, we essentially wanted to zero down on the subjects that had imaging data because we know really well how to do computational neuroscience. We know how to obtain these derived neuroimaging signature vectors. So for these 10,000 subjects, as you can see, 9,900 or so cases, we looked at the highly observed clinical phenotypes, about these 1,500 or so features, and we wanted to examine what is the missing pattern, what features are missing, what is the rate of missing, and as you can see here, there is a very high rate of missingness. So we needed to develop techniques to deal with that missingness. The missingness is obviously not random, as you can see from that pattern. So innovative statistical methods are required to be able to pre-process this specific data set to the point where it can become, again, a computable data object that we can interrogate either with or without model-based techniques. So a little bit about the protocol. So we developed this pipeline workflow that kind of shows on a very high level here the processes that the imaging data goes through, from volume pre-processing to parcellation to computation of the morphometry measures, all the way up to the result aggregation into these spreadsheets or computable data objects. So not everything when you do computing on this kind of scale, not everything is going to be perfect. You're going to have some successes and some failures. So I'm just showing you here that when you do computing on this scale, things are not always going to be right. So in this specific case, we have 99% of the, it says 80,348 compute jobs were done at this specific phase of the pipeline workflow. 99% of them are complete. One was canceled, and 469 instances of the calculations failed. So we can track down. It is very important through the Lonely pipeline environment to be able to track precisely what steps and what cases fail, so that you can remedy and examine if there is anything common for them. Now, obviously, your inference needs to be based on the complete cases kind of going forward. A little bit about the pipeline workflow environment that we've been developing at Lonely and at the INI at USC. This has been a project that, again, has started as being funded as a P41 project, part of the National Institutes of Health. It allows users to design on their client machines these complicated protocols so that the calculations can be outsourced to an external pipeline distributed server where the calculations can be parallelized on complex computational platforms, so that the user doesn't have to worry about managing the data, node assignment, job tracking, aggregation of results, and so forth. This is all kind of managed by the distributed pipeline server on the background. The user only worries about the front-end design of the actual scientific protocol, the algorithm, the pipeline workflow. That's what we do. And then our colleagues at IT and the pipeline team actually worry about all the back-end things that are indeed very important for these types of calculations. So this is a slide that shows you a tool that we developed as part of the Big Data Discovery Science BD2K Center a couple years ago. It's called the Big Data Dashboard. It allows you to essentially query databases, multivariate, multi-source databases, without writing a single line of SQL or any database language code. You essentially visually generate a collage of images, each image representing one or multiple variables. And then all of these charts are interactive, so they're like Trello charts. You can narrow the scope of the states. So, for example, you can select states, or you can select, in this case, this is the histogram of the average commute time for these individuals. So you can manipulate any one of these charts, and as you manipulate one of them, all the other charts morph to reflect the subsetting, to reflect the stratification that the user introduces in one or several of these charts. So during that interactive visual-based data interrogation, you're able to formulate interesting research hypotheses about what might be the multivariate intricate relations that are not a priori known. So you can generate hypotheses that can obviously be tested through rigorous computational mechanisms. So in this example that I just showed you, we used data from the Center for Medicare Services, the CMS, on the Medicare services. We used data from the U.S. Census Bureau and the Bureau of Labor Statistics, just to illustrate how data can be pulled from multiple incongruent, in this case, federal databases, but they're not designed to necessarily interoperate. So we demonstrate how the data can be aggregated and therefore interrogated real-time through visual analytic concepts. All right, so in the last few minutes, I just want to tell you about this compressive big data analytics strategy that our group is developing. So a little bit of motivation here. There is no current mathematical foundation of how to analyze big data. The science, the big data science is not developed. So we have looked at, again, the big data characteristics, and we have introduced this strategy that allows us to do a stochastic-based inference independent of the very specific case-based data characteristics. As long as the data has these 6 dimensions that I showed in one of my earlier slides, large size, coming from multiple sources, incongruency, multi-scale, incomplete, and so forth, we are developing this iterative statistical stochastic algorithm that would allow us to generically process the data in such a way that we generate subsamples from the big data. Then we use model-based or model-free techniques to analyze these samples, compute various likelihoods, estimates, relations, or qualitative or quantitative relations between variables. We repeat this process multiple times, and we effectively look at the asymptotics. We look at the ergodic properties of these algorithms. So this diagram kind of illustrates a little bit of process. It's a 8-step process, very, very generic. It's not, it doesn't deal with brain data. It doesn't deal with neurodegenerative data. It deals with any data set that has the 6 dimensions that I outlined earlier about big biomedical data sets. So the first step is obviously the pre-processing cleaning step, you know, eliminating very strong dependencies, recasting data type when necessary, dealing with missingness, and so forth. In step 2, it does the data harmonization, right? The data set, just like in the Udall, Michigan versus Tel Aviv Parkinson's disease falls data set that I showed you, there has to be a data aggregation and harmonization step. In the second step, we do the variable selection and so forth. And then we go through the iterative CBDA, the Compressive Big Data Analytics iterative protocol, where we randomly sample in step 4 a decent set. Then we do the imputation, scaling, rebalancing, which I mentioned earlier as well when we did the Parkinson's disease study. Then we do the knockoff controlled variable selection, or perhaps other variable selection. It doesn't have to be knockoff. And we apply multiple machine learning methods like the superlearner. Then we go ahead and rank mean, square root errors, or accuracy metrics to identify how consistent are our results across different iterations. And then in the final step, we do the feature mining that allows us to identify these variables that are highly, that appear frequently across the multiple random walks through the cloud of data. So it is a very interesting algorithm. We are right now in the process of validating it. So more on that later. But I want to point out some very important things for us as a community to look at the FAIR data and open science principle. So FAIR refers to findable, A refers to accessible, the I refers to interoperable, and R refers to reusable data set. So as we all collect, manage, process, and understand complex data sets, we should always keep these four in mind, and I'm sure many of us do. This is very important, and the open science aspects, again, not only for the publications, but my group, the SOCR group, is actually using open continuous development. You know, all of these things that I'm showing to you, the CBDA method, it's already on GitHub, even before it's being published. Sharing the data, all the data, the code, and the science is being shared at the time of development. It's not shared after post hoc. It is shared during the process of development. So this obviously entices new investigators to contribute, share, collaborate, extend, and so forth. This final example, I know we're approaching 12:50 here on the East Coast and 9:50 on the West Coast here in the US. So the last thing that I just want to show is another very interesting data set. Now, in this case, about 60,000 patients, and we looked at hospital admissions. The data is not very feature rich in this case. Only 9 features are recorded, including admission, the length of the admission, a binary indicator variable of whether the patient survived, etc., and some demographics. We used very similar predictors trying to identify, for instance, can we figure out who at the time of admission is likely to die, right? So this confusion matrix that you have in the bottom right here, on the bottom left corner, kind of illustrates that the survivals are 9,900. You know, it identifies the patients that the algorithm, in this case random forest, classified as patients that are expected to die versus how many did die. So we have this confusion matrix, and you can see that we have a fairly decent internal cross-validation accuracy of about 85%. The sensitivity is very high, about 93%. The specificity, however, is low, only 13%, which indicates that we have a very, very low rate of falsely positively identifying individual as expected to die when they survived, but we might have a fairly large rate of false negative identification, so people that did die, we might have missed them. On the bottom right corner, we use this algorithm to, on a completely independent data set of 11,000 to 12,000 subjects, to essentially predict the probability of survival. You know, what is that probability? We're not interested in binary classifying individuals as surviving. We just want to have a likelihood which would help physicians, clinicians, and the caregivers. In addition, so this is a completely automated decision support system that adds up another level of quantitative description about likelihood of survival or not. So just to give you, again, coming back to the National Health Service in the UK, you know, we have begun the very same exploratory data analytics using machine learning methods on all of their data. We are developing these workflows. We are looking at their genotypes, and ultimately we're trying to design these connectograms that shows us on the left hemisphere here you have some brain regions or clinical phenotypes. On the right hemisphere you had some of these genomic biomarkers, for instance. Finally, in the interest of time, I'm going to skip this very interesting traumatic brain injury study, because I do want to leave a few moments for questions. You know, the secret sauce of success are 3 things, right? You have to have the funding agencies, you have to have the collaborators, and you have to have the infrastructure to do that type of study. So right up here you have again, recognition of the funding agencies as well as the corresponding grant IDs. Students, fellows, co-workers are very important in this research. Big brain data and predictive health analytics is a very transdisciplinary area. No one can do this as individually or even as in one lab. You have to have colleagues from multiple disciplines, from multiple institutions to do this kind of research. And of course, you have to have the infrastructure, because without the computing resources, without the storage, without the expertise, much of this research is going to be very difficult. So thank you very much. I'll be more than happy to address questions now.
Thank you so much, Ivo. That was an outstanding tour of all these big data analytic projects that you've had going on. One of the things which strikes me before I ask my question, I want to invite members of our audience, if you have any questions, please use the little GoToWebinar question submission system, which is in that panel that's probably on the right side of your screen, to type in your question, and we'll get those texted in, and then we can ask those of Dr. Dinov. Ivo, how important is quality control or some level of quality assurance of these variety of data sets that you're pulling from? In particular, I was struck with all the different publicly available governmental databases that you're pulling from. Do you go through any quality assessment level assessment of that data to go in and make sure that it's valid and make some sense? And how do you look for outliers and that sort of thing?
Yeah, that's a very good question and a very common question, Jack. And I'll tell you, for the dashboard example that I illustrated with the CMS, Bureau of Labor Statistics, and the Census Bureau, we did go through a very manual, labor-intensive quality control. For instance, just to give you an example, there were millions of records, and the federal government databases are full of mistakes. About 3 to 5% of the data was wrong. We were able to correct about 2 to 3% of them simply by cross-validating, for example, things like FIPS codes and zip codes and locale and whatnot. So there is mechanisms that you can do, well, I don't know, semi-intelligent quality control. So that's on one level, right? So there are mechanisms, and you can do this. On a larger scale, however, this process is not practical. And there is a danger. There is a danger that if you collect a big data set, it could represent a very substantially biased or non-representative data. That's always going to be the case. Now, the hope is that as we collectively, as community, improve the data quality, there will be mistakes, but these mistakes on average over the distribution of the data, they're going to be representative of the population. So my personal approach is that we try to do quality control the best we can, as long as it doesn't overwhelm us initially. And secondarily, we are hoping, and we are to some extent validating, whether or not the data is representative of the census. And that's a difficult challenge, without a doubt.
So we have a question from one of our audience, and I was particularly interested too, is you were going to discuss how you use predictive analytics in traumatic brain injury patients, and I was hoping to see a little more description of that before you skip through it, and it looks like somebody else from our audience was too. Can you take a second to just kind of summarize that for us?
Right, absolutely. So let me talk over this a little bit, and there will be a video, I hope, that plays. Right. So traumatic brain injury is incredibly interesting and incredibly challenging. The basic challenge is the heterogeneity of the morbid condition. Right. Everybody can imagine that every single traumatic brain injury is different. So in this video here, we kind of illustrating how we collected data from multiple sources, from multiple individuals. We have access to the infrastructure, the pipeline computing server that you see, and the pipeline graphical workflow environment. And now what we try to do is we literally individually processed every single subject. So we parcellated the brains, we looked at the morphometry metrics that we can automatically compute for every region, for every subject. We're able to reconstruct their cortical surfaces. And not only that, oftentimes the injury is very important, right? Is the injury in the gray matter? Is it affecting the myelinated white matter tracts? How severe is it? Quantifying some of these things as well as the visual representation are very important. And TBI needs to be done on an individual case. So look what's happening with this specific as this image is going to shift now a little bit, you can see that the injury of this patient was in the inferior temporal lobe. Can everybody see the huge gap that we have in the cavity that's indicative of exactly where the injury is? How large is it? How complex is it? So these things are very important. And then we try to look in this specific case, we try to see if the type of injury, was it a gunshot, a motor vehicle, or bicycle accident? And unfortunately, we only have about 40 to 50 cases in this case. We were trying to identify specific commonalities. We did not have enough power to identify anything critical, but the process that we went through was very powerful because we have built the protocol. We know how to analyze these cases. We just need additional, obviously, data and more processing power to be able to obtain these on a larger scale, because without hundreds to thousands of cases, the heterogeneity of TBI is really limiting the scope of any method, model-based or model-free, to yield anything substantially interesting.
That's interesting. You know, we have a question from one of our audience asking about that in the big data era, this era of gathering ever more larger data sets, one of the first things people seem to be interested in doing is applying machine learning techniques in order to be able to make predictions about, you know, classifying subjects, or being able to make predictions about you know, what something might be, or whatnot. But the alternative is, can we make inferences? And how do we make inferences even though we can make these algorithms which do prediction? And is there a distinction? And how can we identify features or the relative importance of some variables versus others? And I think you're uniquely qualified to comment on this, so I thought I'd throw this back to you.
Right, so that's a very interesting point. I want to start off by saying that data science is recognized effectively by many of us as the fourth scientific pillar, in addition to the experimental sciences that started during, you know, well, during the Renaissance era, to the theoretical sciences that started, you know, in the 17th, 18th, and 19th century, the computational sciences, which effectively matured in the 20th century. In the 21st century, we have the data sciences. It is true that you can, scientific discovery can be hypothesis-driven or exploratory. So this can be called exploratory versus confirmatory science. Machine learning methods can be used for both. You can use machine learning methods to formulate a research testable hypothesis that can be prospectively tested or validated using other methods, but it can be used for inference as well, simply because the statisticians in the past decade have realized very interesting phenomenon. Okay, hypothesis-based inference is rooted in the notion that you quantify uncertainty in terms of p-values. Data science inference is rooted in the understanding that the probability values that you compute here are replaced by or augmented by looking at quality metrics like the log odds ratio, like the positive and negative predictive power, like the sensitivity and specificity on a statistical cross-validation world. So in other words, we pull the data where every element can be used, or every case can be used both as an element that we learn from and as an element that we can assess the predictive power of these algorithms. So it is very interesting. It's a very rapidly evolving field. Obviously, there'll be mistakes. Let me remind you of the Google Flu Trends fluke. You know, things are not going. Some things are not going to work well. Some things will work well. And I know I'm out of time, so I'll stop here. But any other questions or quick ideas, Jack?
I think we've run out of time, but I do hope that our audience will feel free to find Dr. Dinov at the University of Michigan and shoot him an email should you have any additional questions for him. I'm sure that he'd be delighted to answer them. But I hope you'll also join me in thanking Dr. Dinov for his presentation on Big Data Brain Science and Predictive Health Analytics for our BD2K Guide to the Fundamentals of Data Science. Thank you so much, Ivo. It was an absolute pleasure. And I want to thank our audience for taking the time today to join us. I want to wish everybody a happy holidays, and do watch for, this will be our last data science seminar for 2017, but start watching for a new collection of these to begin shortly after the new year. Once again, thank you everybody, happy holidays, and my best to you and yours for the remainder of the year. Anyway, thanks so much again.
It was a pleasure. Thank you, Jack.